Hey everybody, it's Dr. Rindy. Welcome to the next episode of WIALD, What I Am Learning Daily, where you are welcomed into my personal learning lab. This particular week, I'm focusing on a presentation I attended as part of the Microbiome Summit that's being hosted by Lucy Mailing, Dr. Lucy Mailing. I want to give her a quick shout out because she's doing some amazing stuff with microbiome research. And this particular lecture that I attended had to do with one of my favorite topics, low fiber versus high fiber diets and its impact on inflammation in the gut. This was done by Dr. Martens of the University of Michigan. He conducted this research on rodents. However, it had some interesting takeaways to help clinically. So first, some backdrop on the microbiome that I think is relevant to this lecture. Bugs feed on all types of nutrients. They can feed on fat. They can feed on carbohydrates and proteins, and also can feed on mucin, which is mucus from the intestinal lining or mucus that's produced. So it's important to know that we have a group of microbes that are called mucinophilic. That's pretty much one of the focus of this particular lecture. If there's too much mucus degradation, it can kind of go into the intestinal lining. However, some mucus degradation can be appropriate and helpful in certain scenarios. So let's go into what we learned. So two of the species that were discussed that are mucinophilic are Bacteroides casei and Akkermansia mucinophilia. There's more you can learn about these particular bugs, but I think the important takeaway is both of them feed off mucus. And Akkermansia mucinophilia is widely talked about in the research. I've seen a lot more research on its benefits than versus its harm, and as its benefits to control inflammation. Also, it kind of has been shown to help with certain disorders such as allergy disorders and IBD and IBS. So it was a little surprising that that was the focus of this particular study. However, that's the thing with microbiome research, is that it can kind of teeter-totter back and forth with some of these bacteria, and to understand their benefits, risks, and the balance of these microbes in our ecosystem. So the researcher, Dr. Martens, it was kind of set up for his research by another study by Dr. Desai, who published an article in Cell in 2019. They did a low fiber diet in mice, and they looked at what would happen to the microbiome population if you held the fiber for 40 days or so. And not surprisingly, holding the fiber raised up these mucin degraders. They grew. But also, we also saw that a pathogen was elevated in this particular mice population of wild-type mice called Citrobacter rhododendron. So there was a pathogen that came along, and that was an interesting finding. So the research took this kind of concept and they applied it to an IBD model. And in IBD, there's many genetic pathways that have been discussed to be associated with IBD that are related to immune regulation. One of them is FUT2, which we talk about a lot with intestinal lining. The researcher, Dr. Martens, talked about one called cosmic, which is involved with intestinal barrier degradation. But one of the most common pathways that's looked at is something called interleukin 10 deficiency, which is an immune regulation pathway that seems to be associated with IBD. Let's unpack IL-10 just a little bit more. IL-10 deficiency is sort of the most common genetic model used in studying IBD. They look at IL-10 loss as being part of the IBD picture, inflammatory bowel disease, with low IL-10 production can be sort of a scenario where we would see the autoimmune flare up. So IL-10 is made by T regulatory cells. And so one thing to know about IL-10 is it dampens the T effector cells. And T effector cells are Th1 and Th2. These are T helper 1, T helper 2. These are largely associated with inflammation. So we see the classic TNF alpha, interferon gamma go up, and other cytokines that are associated with inflammation when Th1 and Th2 are active. So as you can see, if you don't have IL-10 working real well, it can really set you up for autoimmunity, where the body is sort of the immune system is attacking itself. So in this experiment, they took IL-10 mice and took IL-10 deficient mice. They knocked out IL-10 out of these mice and gave them a high fiber diet versus a fiber-free diet. In the low fiber-free diet, the mice eventually lost too much weight and became fatal. They died from weight loss, and then also the immune system went up in these mice, immune markers of neutrophils in a fiber-free diet. And the mice that got sicker on this diet had the classic cytokine profile that we see with IBD. So they had elevated IL-1 beta, IL-6, IL-17, IL-22, TNF alpha, and INF gamma. So you can see that this, without IL-10 and low fiber, that was a very kind of difficult, fatal combination to show the amount of colonic inflammation that developed in these IL-10 deficient mice. So then they moved on to a kind of interesting side study where they looked at if they were to give the IL-10 deficient mice something, what's an exclusive enteral nutrition diet. This is sometimes known as the elemental diet. It's devoid of fiber, and they wanted to see what would happen in these mice for inflammation control, because the EN diet has been shown to help with some humans with IBD and SIBO, specifically like Crohn's and also pouchitis. It's been shown to help. And so they looked at what would happen if a fiber-free diet was given to these IL-10 deficient mice in the form of the elemental diet. So what happened was interesting. One of the butyrate producing bugs actually grew, Eubacterium rectale, which is a butyrate producer, butyrates should only be produced in the presence of fiber. But it was interesting and unique that Eubacterium rectale grew in the presence of no fiber. So I'm going to come back to talking a little bit more, but I want to unpack about Eubacterium rectale. It's a member of Clostridium cluster 4. Clostridium cluster 14A. That's a really dominant group in the intestinal microbiota. It has many different species as a part of that cluster. Some of the Clostridium species, Ruminococcus navis, is also represented in that group. But as I learned about this, I saw in a particular study by that was produced in this article called Biodiversity of Intestinal Lactic Acid Bacteria in Healthy Populations in 2016 in the publication Advances in Experimental Medicine and Biology, that these particular bugs, especially it looked like Eubacterium rectale, could actually survive on protein, and that some of the Clostridium cluster 14A can actually eat protein as nutrition. So perhaps that's what was happening. I'm too new at that particular question to conclude that, but that's sort of my initial hunch. The researchers did not say that butyrate was produced. So if butyrate was produced on the EN diet, that would be really fascinating, but that was not pointed out as far as I can recall. So that's my answer to why possibly Eubacterium rectali it grew on that diet. So what happened in this study when they refeeded these mice that were IL-10 deficient with fiber? Well, one thing that was up in these particular mice while they were flaring was fecal lipocalin, which is a marker of intestinal inflammation, and it remained elevated while fiber was being added, but then it slowly decreased. So this is a really good clinical pearl for me. Is, the switch takes time. If you're kind of adding nutrition into a diet, like say someone's flaring and they're holding off nutrition, it takes time to see inflammation come down with fiber introduction. So, but in these particular mice, they added oat fiber, wheat fiber, and the disease started to blunt. So, Dr. Martens discussed how that perhaps a low fiber fingerprint is elevated Akkermansia muciniphilus, and the Bacteroides casei, maybe if they're elevated, that could be a low fiber fingerprint. I'm sort of, in humans, I'm not quite sure I see that with the stool test that I do, but it is an interesting concept. And also I can only imagine that the longer we avoid fiber, the more these bugs really become elevated and potentially could cause problems like intestinal permeability, even in people without IBD, in IBS patients or general populations. So it does bring to me some questions as to how much Akkermansia muciniphila is beneficial, and along those lines, how much Bacteroides casei is beneficial, and under what circumstances are they helping us and hurting us. And again, I'm not 100% clear why butyrate producers would grow in the absence of fiber, but I'm guessing that at least they can derive some of their nutrition from protein. And what would happen if people were starving themselves of fiber for 6 months or more? That's a big question on my mind. And, so the kind of, as a naturopath or functional medicine doctor, I'm kind of always looking at balance. And what I take away from a study like this is what's true in a short-term stressor might be that the body may try to bring homeostasis to an environment, and we might see some benefits from certain microbes, but long-term stressors, those particular microbes might turn against us. So it's really important to always think about timeline and sequencing in our clinical thinking, and just try not to think about things as like a snapshot in a moment, but to think about, well, what would happen as a rhythm over time in this scenario. So that's my takeaway. I really enjoyed this lecture, and I'm enjoying sharing my learning with you. So please tune in next time for the next episode of WIALD.